There is a process running inside your eyes right now that has nothing to do with what you are looking at. It has nothing to do with light, color, or focus. It is a maintenance system working in the background of every single cell in your retina. And most people go their entire lives without knowing it exists. Scientists have spent decades mapping what happens when this system is pushed to its limits. Specifically, what happens after 96 hours without food? The answer involves your optic nerve, your retina, and a biological mechanism that may be quietly deciding how well you will see decades from now. Every cell in your body generates waste as a natural consequence of staying alive. Proteins fold incorrectly. Mitochondria wear down. Cellular debris accumulates as a byproduct of simply functioning, and nowhere is this more relevant than inside the eye. The retina is not a passive screen receiving light. It is one of the most metabolically demanding tissues in the human body, and that demand comes with a cost that has to be managed continuously. This management happens through a process most people have never heard of, despite it operating inside their eyes every second of every day. It does not pause when you sleep. It does not pause when you are reading this sentence. It is a constant low-level cleanup operation that determines whether cellular damage gets cleared out or allowed to pile up over years and decades. This story gets genuinely interesting once that system is pushed far beyond its normal operating range, past 72 hours, past the point where most people have ever gone without eating, into territory researchers are still actively mapping. Extended periods without food do not simply pause ocular function. They appear to trigger a specific, measurable shift in how retinal cells handle their own maintenance. And by the end of this video, you will understand exactly what that shift involves. The term comes from Greek, combining auto, meaning self, with phagy, meaning eating. Autophagy is, quite literally, a cell eating parts of itself, though the process is far more controlled and purposeful than that description might suggest. Inside every cell, including the cells that make up the retina, there is a system for identifying damaged components, wrapping them in a membrane, and delivering them to a structure called a lysosome, where they are broken down into reusable molecular parts. This discovery did not emerge overnight. Japanese biologist Yoshinori Osumi spent years mapping the genes responsible for this process in yeast cells during the 1990s, work that eventually earned him the Nobel Prize in Physiology or Medicine in 2016. What he uncovered turned out to apply far beyond yeast. The same fundamental machinery operates in human cells, including the highly specialized neurons that make up the retina. In the eye specifically, this cleanup system has an enormous workload. The retinal pigment epithelium, a layer of cells positioned just behind the photoreceptors, is responsible for processing roughly 30,000 shed photoreceptor segments every single day. That number alone explains why autophagy is not optional background maintenance in the eye. It is a structural necessity for the tissue to keep functioning at all. Photoreceptors, the cells responsible for converting light into the electrical signals your brain interprets as vision, carry an unusual distinction. They contain a higher density of mitochondria than almost any other cell type in the human body. Mitochondria are the structures responsible for producing cellular energy, and the sheer concentration of them inside photoreceptors reflects just how much fuel these cells burn to keep up with constant visual demand. This is not a passive process. Photoreceptors regenerate a portion of their outer segments daily, rebuilding the very structures responsible for capturing light. At the same time, they maintain something researchers call the dark current, a continuous flow of ions across the cell membrane that consumes a striking share of the eye's total energy budget, particularly in darkness. None of this happens without enormous metabolic cost. High energy production comes with a trade-off that applies to every engine, biological or mechanical. byproducts accumulate. In photoreceptors, that means oxidative stress, damaged mitochondria, and misfolded proteins building up as a direct consequence of the very process that allows vision to function in the first place. Autophagy exists specifically to manage this trade-off, clearing out the damage before it accumulates to a level that compromises the cell. Without it, the retina's own energy demands would eventually work against it. When the body stops receiving food, cells throughout the system begin shifting their energy strategy. Glucose, the body's primary and most readily available fuel source, becomes less abundant, and cells must adapt to function with less of it. This shift does not happen uniformly or instantly. It unfolds in stages, and the eye experiences this transition alongside every other tissue in the body. Research conducted on retinal tissue during fasting has shown that this nutrient shortage acts as a direct trigger for autophagic activity. A study examining ischemic retinal injury found that fasting periods of 24 to 48 hours produced a measurable, sustained increase in autophagy markers within retinal ganglion cells, the neurons responsible for carrying visual information from the eye to the brain. This activation was not incidental. It corresponded with improved survival of these same neurons under stress conditions. This early window matters because it establishes the foundation for everything that follows. The cellular machinery responsible for autophagy does not switch on like a light. It ramps up progressively as nutrient scarcity persists, and the first 24 hours represent the opening phase of a process that, according to the available research, continues to evolve well beyond this point. By the 48-hour mark, the picture inside retinal tissue begins to look different from the initial response. The same research examining retinal ganglion cells under ischemic stress found that fasting periods extending to 48 hours did not simply maintain the autophagic activity seen at 24 hours. They sustained and prolonged it, extending a response that, without continued nutrient restriction, would have already begun to taper off. This distinction matters scientifically. In the study's non-fasted comparison groups, autophagic markers in stressed retinal tissue peaked early and then declined within roughly 24 hours as the cellular machinery responsible for the process became exhausted. Extending the fasting window changed that trajectory entirely, keeping the autophagic response active well past the point where it would normally have shut down. What makes this finding significant is the outcome attached to it. Sustained autophagy at the 48-hour mark was directly associated with improved survival of retinal ganglion cells following injury, suggesting that the timing and duration of this cellular response carries real biological consequences for the neurons responsible for vision, not just a passing shift in cellular chemistry. Those same retinal ganglion cells just described, the ones whose survival improved under sustained fasting-induced autophagy, sit in a uniquely vulnerable position within the visual system. They are the neurons responsible for collecting visual information processed by the retina and transmitting it via long axons bundled together to form the optic nerve, all the way to the brain. Because they are neurons rather than constantly regenerating epithelial cells, retinal ganglion cells cannot simply be replaced once lost. Whatever damage accumulates inside them over time tends to stay, making their internal maintenance systems disproportionately important compared to more replaceable cell types elsewhere in the body. This is part of why researchers studying neurodegeneration have focused so heavily on this particular cell population when trying to understand how cellular stress translates into lasting visual consequences. Autophagy inside these cells operates as a quality control checkpoint, continuously identifying and clearing damaged mitochondria, misfolded proteins, and other compromised components that would otherwise interfere with the cell's ability to conduct electrical signals across long distances. Research using optic nerve injury models has documented that autophagy increases rapidly and consistently following nerve damage, and that blocking this autophagic response with pharmacological inhibitors significantly worsens axonal degeneration. This finding established something important. Autophagy in this specific neural pathway is not merely present as background maintenance. It appears to play an active, measurable role in determining how much damage the optic nerve sustains after a stress event. What sustained nutrient restriction does to this system is layer an additional variable on top of an already delicate balance. The molecular signaling pathway most responsible for regulating autophagy, centered on a protein complex called mTOR, responds directly to nutrient availability. When food intake stops for an extended period, mTOR activity drops, removing one of the primary brakes that normally keeps autophagy in check. Research examining human skeletal muscle after a 72-hour fast documented a roughly 50% reduction in mTOR phosphorylation, alongside reduced signaling through ULCOM, a protein directly responsible for initiating the autophagic process. While this particular measurement came from muscle tissue rather than the retina, the same fundamental signaling pathway operates inside retinal ganglion cells, which is precisely why researchers studying ocular autophagy have looked so closely at extended fasting windows as a tool for understanding how far this system can be pushed before its behavior changes. By the time a fast reaches the 72-hour mark, research on human skeletal muscle has already documented substantial shifts in the signaling pathways that govern autophagy throughout the body. And there is no biological reason to assume the eye operates in isolation from this broader metabolic shift. This is the window where fasting research intersects most directly with one of the most heavily studied areas of ocular autophagy: glaucoma, a disease characterized by progressive degeneration of retinal ganglion cells and their axons, frequently linked to elevated pressure inside the eye. The relationship between autophagy and glaucoma, however, is considerably more complicated than a simple story of cellular cleanup preventing disease. Multiple studies using animal models of chronic ocular hypertension have found that autophagy markers rise progressively as elevated eye pressure persists over time, with one study documenting a sustained increase in autophagy-related proteins over 8 weeks of chronic pressure elevation. Separate experimental work using pharmacological autophagy inhibitors in glaucoma-related injury models found that blocking autophagy reduced the rate of retinal ganglion cell death, a finding that runs directly counter to the assumption that more autophagy automatically means better outcomes for the eye. A genetic model targeting autophagy genes directly reached a similar conclusion, finding that autophagy deficiency protected against both ocular hypertension and the neurodegeneration that typically follows it, leading those researchers to describe autophagy activation as playing a potentially harmful rather than protective role under those specific high-pressure conditions. This complexity matters enormously for how this story gets told honestly. The relationship between extended fasting, the autophagic activity it triggers, and intraocular pressure is not a straight line where more cellular cleanup simply equals a healthier optic nerve. Context determines outcome. The same biological mechanism that helps clear damaged retinal ganglion cells following an ischemic injury appears under chronic high-pressure conditions associated with glaucoma to potentially contribute to the very cell loss researchers are trying to prevent. Untangling exactly when autophagy helps the eye and when it works against it remains one of the genuinely open questions in this field of research. And 72 hours into a fast sits right in the middle of the window where this question becomes most relevant. Reaching the 96-hour mark places a fast well beyond the range most metabolic research has historically studied in humans, which is part of why this specific window remains scientifically interesting rather than fully settled. The most directly relevant human research available comes from a clinical trial design examining 3 full days of water-only fasting. measuring autophagic flux in blood cells alongside a wide range of other biological markers, including genomic, proteomic, and metabolomic changes. This represents one of the only attempts to rigorously quantify what extended fasting does to human autophagic activity in real time, though its measurements come from circulating blood cells rather than ocular tissue directly, a distinction worth holding on to rather than glossing over. What the broader fasting and stress response literature does establish is that autophagy unfolds in recognizable phases rather than as a single continuous escalation. Research on cellular stress responses describes an initial phase where autophagy is induced within minutes to seconds of a stress trigger, followed by a second phase where, if the stress persists long enough, additional pathways become active, including processes connected to protein misfolding responses and, eventually, programmed cell death. This two-phase structure helps explain why scientists treat extended fasting windows like 96 hours as scientifically meaningful checkpoints. At some point, a process that begins as protective cellular maintenance can, under sufficiently prolonged stress, start to overlap with pathways connected to cellular damage rather than repair. Applied specifically to retinal tissue, the animal research on ischemic retinal ganglion cells offers the closest available parallel. Sustained fasting in that research kept autophagic markers elevated well past the point where they would have otherwise declined. And this sustained activity corresponded with improved neuronal survival following injury, at least under the ischemic stress conditions being tested. Whether that same pattern of benefit continues, plateaus, or reverses at the 96-hour mark specifically has not been directly measured in retinal tissue, human or animal, which is precisely the kind of honest gap that separates established science from speculation dressed up as certainty. The two-phase model of autophagy under stress points towards something biologically significant. The same machinery responsible for clearing cellular damage can, if nutrient restriction continues long enough, begin drawing down resources the cell needs simply to keep functioning. Autophagy does not run on nothing. The entire process of forming autophagosomes, fusing them with lysosomes, and breaking down their contents requires energy, meaning that a cell sustaining high autophagic activity over multiple days is spending real metabolic resources on its own maintenance. At the same time, those resources are becoming progressively scarcer. Beyond muscle tissue, research on whole body metabolism during extended fasting has tracked a broader shift in fuel strategy. As glucose and glycogen reserves run low, the body increasingly relies on fat-derived ketones and, eventually, on breaking down selected proteins to sustain energy supply. This broader pattern, not limited to any single tissue, illustrates the same underlying principle. Multiple systems within the body shift simultaneously as nutrient scarcity persists, and cellular maintenance processes do not operate in a vacuum separate from the rest of the body's metabolic state. For neurons specifically, including retinal ganglion cells, this tension carries particular weight because these cells cannot be replaced once lost. A cell that successfully uses autophagy to clear damaged mitochondria and misfolded proteins during extended fasting may emerge from that stress period in better condition than one where autophagy was blocked or impaired, based on the ischemic injury research already discussed. But a process pushed past the point where it can be adequately fueled risks tipping from protective maintenance into a contributor toward the same cellular dysfunction it originally evolved to prevent, a balance that appears to depend heavily on the specific type of stress involved, the cell type in question, and a duration threshold that current research has not yet mapped with precision for the human eye. Pulling these findings together reveals a picture considerably more nuanced than a simple story of fasting cleaning out the eyes. Autophagy inside retinal tissue functions as a constant necessary maintenance system, one that processes tens of thousands of photoreceptor segments daily and manages the oxidative byproducts generated by some of the most metabolically demanding cells in the entire body. Extended fasting clearly influences this system, shifting it through identifiable phases as nutrient availability changes. And research on retinal ganglion cells facing ischemic injury has shown that sustained autophagic activity during fasting correlates with meaningfully better neuronal survival under those specific stress conditions. At the same time, the glaucoma research introduces a genuine complication that responsible science communication cannot skip past. In the context of chronic elevated eye pressure, the same autophagic process that helps neurons survive acute injury has, in some studies, appeared to contribute to the very neurodegeneration researchers are working to prevent. This is not a contradiction in the data so much as a reflection of how biology actually works. The same cellular tool can serve different functions depending on the specific stress it responds to, the duration of that stress, and the broader physiological context surrounding it. What ties all of this together is a relatively young and still developing area of research. Human studies measuring autophagy directly in ocular tissue during extended fasting remain extremely limited, with most of the field's foundational findings coming from animal models and, more recently, blood-based measurements in ambitious human fasting trials. The 96-hour mark sits at the edge of what current science has mapped with any precision, which is exactly why researchers continue to study it. Not because the answers are settled, but because the question of how far this ancient cellular cleanup system can be pushed, and what that means for one of the body's most energy-hungry tissues, remains genuinely open. If this kind of deep dive into the science behind your own body is something you want more of, the best thing you can do is hit that subscribe button, because there is a lot more of this kind of research waiting to be unpacked. And if there was one part of this video that genuinely surprised you, drop it in the comments below. I read every single one. If this helped you understand your own eyes a little better, share it with someone who would find this as fascinating as you just did. I will see you in the next video.